opening that, I'm going to jump to the protein group file. So this is only made if you do protein inference. And so post-protein inference, we create proteins and protein group identifications, and those are also then mapped back to the PSM and peptides file to make sure that everything like corresponds. So we have the protein accession. So here, we are able to determine that this single protein is in our sample, or this is how we display protein groups. So these 3 protein accessions, we can't distinguish based on the peptide evidence that we have, if one is in the sample, if it's a subset, or all of them. So we display all equally here. And this is the shared peptide support for that. So here we give our protein accession or the protein group accessions, gene, organism, names, the mass of the unmodified protein, the number of proteins in group, each group. So this is a nice way to kind of filter there. And then we tell you which unique peptides we've identified for that protein and which shared peptides we've identified. Total number of peptides, number of unique peptides, and then sequence coverage fraction. So we say, based on the peptides that we've identified for this protein, what percentage of all amino acids do these cover? So that here, and then here's some like kind of way to visualize that. So we have sequence coverage here. Lowercase are that are not covered. Uppercase are ones where we have peptide support for that. It gives it as a base sequence. Also, if there's modifications, it includes that here. Gives the intensity for that protein, the number of PSMs for that specific protein, whether it's a target or decoy, the best peptide score for that protein. And then we do a protein level FDR that is kind of based off of the top picked approach. So, which essentially means that if you have a target protein group and a decoy protein group, and the target outscores the decoy protein group, you keep the target protein group, which helps not be overly conservative in the estimation of protein level FDR.